Here also 100% of the glucose and 100% of the amino acids are also filtered. Now when the fluid reaches here, it constantly loses the solutes and the water follows by the solute, and this is also known as the solvent drag. This part of this whole part, this is water permeable, but the thick ascending part of loop of Henle, thin ascending part of loop of Henle, and small part of DCT, this whole part is watertight or water impermeable. As the solute reaches here, then it goes upward. Here the solutes are freely reabsorbed into the circulation, but the water doesn't follow because this portion is watertight. Here are in this part, that is the thick ascending part, there are pumps. This is known as the located transporter or pumps, you can say, that is sodium potassium 2 chloride pump. And this is called this one is the luminal side. This is the basolateral side. On the basolateral side, here are present sodium potassium ATPase, which are ATP dependent, energy dependent ATPase. The sodium which is reabsorbed from the thick ascending part of loop of Henle, it goes to the general circulation by this pump. It goes out by the sodium potassium ATPase. When 3 sodium goes out, 2 potassium reach inside the cell. Here potassium also goes inside the cell, so the cell becomes potassium rich and sodium poor. Now, and there are also located some chloride channels here. So the chloride which enters from the tubule into the cell, it goes out into the circulation by these channels. It goes out, but the cell becomes very rich in potassium. Now calcium has to leak back. So on the luminal side, there are certain channels present which are known as potassium leaky channels. Potassium leak outs here. Now here becomes, now this part of the thick ascending part, this part becomes highly electropositive. Here there are also present certain calcium and magnesium ions which also have to escape out. These, in order to prevent such high electropositivity here, some of the calcium and the divalent ions, calcium and magnesium, these are divalent ions, these rush out through the junctions present. These junctions are known as tight junction. So these are reabsorbed in this way by the high electropositivity here. So these solutes are thrown out into the interstitium, but water doesn't follow by. So in this way, the medullary interstitium becomes highly hyperosmolar because this all part is watertight. It doesn't allow water to follow the solutes. Now here, now again, And the last part of the nephrons, the collecting ducts, this all part, this is dependent on ADH for the urine concentration and dilution. In the presence of ADH, this part is water permeable, and in the absence of ADH, this complete part is water impermeable.